He's the administrative coordinator of GHGA. So please, Jan.
Yeah, thank you, Anna. Thanks for opening. I just posted you had the wrong date for Sergei Beltran. It was actually today. Was the date? It's on October 24th. The next lecture. Yeah, but today William Rayner is going to present his talk in the GHGA lecture series. Will is head of data informatics of the data informatics group at the Institute of Translation and Genomics from Helmholtz Munich. So Helmholtz Munich is also a partner organization in GHGA, although that group is not directly part of GHGA. Will holds a degree in biochemistry and later a PhD on computational protein structure from Leeds. I think that's also some time ago. After that, he had a lot of placements first in biotech startups, and then he worked for quite a long time in Oxford at the Wellcome Trust, and I think also at the university in different data management positions as a senior bioinformatician, where his main research focus was type 2 diabetes. And at that time, I mean, many new technologies have developed, starting with GWAS. So he was involved in a lot of GWAS projects, developed tools for that, and also was part of many consortia over the time. One consortium he's today part, and he's going to talk about that, is a CINECA project, which is actually a global project, which you actually hear quite often. So I just returned from the Global Alliance for Genomics and Health meeting, and there also were presentations on this project, which is really very close also to what GHGA wants to do, namely share data across borders in a secure way. And federation is a term often heard from that. So we are really looking forward to this presentation from Will for first-hand experience for use cases within CINECA. And yeah, Will, please share your screen, and we're looking forward to your talk.
Thank you for the opportunity to present the work here. And I hope this will be sort of an interesting sort of overview in the work that we've been doing in sort of CINECA project and sort of associated with that. So I'm going to go into a bit of sort of background, sort of some of my background, some of the background around sort of federated systems, a bit of an introduction to CINECA. Apologies if you've seen this before. And then I'm going to talk about the work that we've been doing within CINECA in one of the use cases there, and sort of highlight what we've done, what we found useful, and sort of, as I said at the end, our lessons learned. So, as has been commented, my background is that I'm sort of really interested in everything around sort of data management, data privacy, data security, and the focus is generally on sort of genomic and sort of associated phenotype data. I've not long been at Helmholtz Munich, and I'm still getting to know the place because obviously moving jobs during a pandemic is tricky. Prior to that, I did spend a very long time at Oxford University. There I was within the type 2 diabetes group, and that was the predominant phenotype, but there were a lot of other metabolic diseases and some type 1 data diabetic complications. And I was ending up managing data on sort of 60 or 70 different cohorts of data, and being able to deal with those as the new technologies came along. And prior to that, I'd worked at a couple of small biotech companies in the Oxfordshire area, again, sort of on data and database management. So I've really had sort of a long history dealing with data. And I guess the question is, I probably don't need to state this, is why are we doing what is federated data analysis? And it's the idea is that for us here, is you can analyze data on multiple data sets that reside on different computer systems, potentially within the same country or around the world, as if they were all residing locally. And this is equivalent to effectively what the work we've been doing in large consortia have been doing for their meta-analyses. And on the right here, you can see the different cohorts. You would meet these people, you either knew them or whatever, and they would run the analysis for you on their raw data. they would then send you some summary statistics, and then you would combine these summary statistics in a final meta-analysis for your final discoveries. And these have worked exceedingly well. There are large numbers of loci have been discovered in common complex diseases, but this can be very slow. You're reliant on analysts at the other end having time to do this and to do the work and send you back the results and hopefully send them correctly. So why? I mean, obviously you would like to automate this more. Well, speed, obviously it takes a long time to get these people to do the work. The data size, and I don't need to sort of highlight that this was a slide put together quite a few years ago now on the number of large-scale human cohorts that were being planned then. This was sort of prior to CINECA starting, and sort of highlighting the need as why we're going to need some way of being able to link up these data sets because of the sheer size and distribution across the globe, and shipping sort of data sizes of petabytes like the UK Biobank now is around the place is very inefficient. And I think there has been a massive increase in the awareness around sort of privacy and privacy of people's data, especially the new GDPR regulations, and the case they make for genetic data being a special case, and the restrictions around what you can actually do with it, how you can share it, and how you deal with personally identifiable information in that case, given that genetic data not only identifies an individual but obviously the whole immediate close family as well. And we're ending up with effectively what is going to be a number of data, what I call silos. These going to be sort of trusted research environments like the UK Biobank have done, or things like the European Health Data Space. And I'm going to take a quick diversion off into that just for the moment, for those of you who might not be aware of this, but this is something that is being planned by the EC in Europe for different data spaces within different areas. The health is the first. The idea is that individuals will actually have control who has access to their health data. So if you move doctors, you can rescind the permission from one and add it to another. If you're abroad, you can grant it to someone else. If you want to see another doctor in another country for something else, then you can do that as well. And so this will allow for sort of EU-wide storage of electronic health records and associated data. And the data will be stored in one or more potentially data centers in each country. And the legislation for this includes the possibility of using the data for research as a secondary use. And given the increase and amount of health data and sequencing being done for that, this will be really key to accessing this data and utilizing it for driving future research and discoveries to benefit the health of people down the line. So these are slides from the EC in their latest presentation with the release, and effectively on the right here you have all the different types of data. They're going to create standardized metadata on this, put it in a catalog that you can search via a trusted portal, and this will all be managed by these health data access bodies, of which there will be one or more per EU country. And the idea is that you will use that portal to discover data, then you will put in an application, and assuming it's successful, then they will prepare the data and provision it in a secure environment for you where you can analyze and process the data. And this, I believe, is kind of starting towards, again, another sort of slightly federated system potentially. The idea here being that this triangle in the right is sort of on a national context, but these health data access bodies would be per country and they would be interlinked. And if you put in a request for data EU-wide, it would be across all of these health data access bodies, not just one, and you would only have to apply to your local one to get the data. And there would be a lot of other environments like the secure processing environments and facilities provided by different national bodies, but also central support services. And so they have this lovely slide, which is rather complicated in terms of how the data would be, data flows would happen. But I think the main thing is that, again, you're searching the data. This is a bit like the GWAS. You search for the data, you find the data holders, you request the data from them, then you have the data available to analyze, and you can then take it back, or this exporting anonymous data only here. So you have this access to this environment, and these are all time limited, so that once you've done it, then things will be deleted after it's all been sorted. So I started off working really with Federated Systems, and this was work done with the people at EBI, with Dylan, to help work on the federated nodes for the knowledge portal, which was the type 2 diabetes one, which is now sort of the common metabolic diseases portal. And this was specifically to allow data that couldn't be deposited in the US to be included in the portal for analysis and integration with the main data sets. And from there, then I moved on to CINECA, which is going to form the bulk of this talk. And this is really looking at infrastructure in Europe, Canada, and Africa, and trying to set up a demonstrator of the technologies for this, so that we can, as we said in the mission statement, enable large-scale kind of genomic and data, so across borders, so that we can accelerate research and improve health. And the aim really is that, you know, as we mentioned, there are these huge numbers of biobanks, both in Europe and worldwide, but we cannot take the data from them. We're going to have to find some way of working with all the ethical, legal, social implications, the security requirements, the restrictions on privacy, but still being able to deliver on the research. And the idea is that CINECA is going to take these standards and try and create this federated infrastructure. And the whole idea is really this is a demonstrator for this a federated infrastructure. And these were kind of listed as the sort of main challenges for doing this kind of work. One is actually finding the data sets. I mean, when we're doing it as a GWAS, we do that, you know, you end up presenting at a conference and you have 5 more people approach with data sets saying we'd like to join. But once you've got access to, or once you know about the data sets, it's getting access to it. How do you actually do that? So there's got to be some kind of authentication, authorization infrastructure. The data have to be comparable. So this is, you know, we need to know about the metadata and it's consistent across the data sets. And then actually being able to do the federated analysis, and also have a support for the ethical, legal and social side of things. And so this was the structure of CINECA. Obviously, EU grant with work packages, and each work package is kind of matching up to one of those challenges, so that we have work package working on the metadata and the authorization. There's the federated discovery, and 2 working on federated applications, 1 on research and 1 on clinical applications. And the work I'm doing is falling under this work package for federated research. And we are planning to really follow the Global Alliance for Genomics and Health kind of standardized standards. Effectively, they've been put out there as to you know, how we can potentially do this. And so we're going to try, you know, reuse existing technologies, reuse things like Beacon, the ELSI, but existing programs like nextflow, and put these together in such a way that we can actually create this federated structure. So there's a lot of interdependencies on the work packages, and we have a lot of cross work package calls to try and ensure that the data flows as they go through from the discovery and the authorization work well with our access to the data and trying to standardize the metadata as well. And so in Work Package 4, we have several use cases, and we took early on the decision that we wanted to really standardize how these work in terms of the broad framework. And this kind of is exactly the same as the meta-analysis one I showed earlier that we're working on in large consortia, but the idea here being that now we would actually have this more automated, so that we would use our Work Package 1 and Work Package 3 to search and standardize the metadata to identify the cohorts that we need. Then you would actually contact the DACs to get access, and you'd be able to then potentially use the distributed authorization to get access to the data. And with that, they would either potentially, depending on the permissions, be able to either download the data or upload the tool to run to create this summary product that is no longer personally identifiable, which you would then download and then sort of meta-analyze. And the different use cases we decided basically had the same structure. So we're trying to work very closely between the different use cases to come up with the same protocols, but we just plug in different modules for the different use cases. So this comes back to this slide here, I think, from the EC. And again, this I think is showing this is very similar, that you're kind of looking up the data here in a public metadata catalog, you're requesting the data with some kind of authorization, you're getting the data in a secure processing environment where you either upload your tools and export the anonymous data back down for your combination. So it's kind of very similar, and I think this is kind of one of the reasons that we need to drive forward the federated analysis, that there are so many different areas where we'll have data to deal with. So that the use case I've been working on has been this one on polygenic risk scores. So we decided that this was kind of a very interesting area when we started a few years ago, or 3 years ago now. And at that point, PRS was starting to be used, but generally the idea was you would require some access to individual level data. So our initial stated aim was that we would be able to take a PRS that we had generated or downloaded from the PRS catalog, and that we would share the variants that comprise that with our workflow, and that would then be available to run on all these different cohorts to evaluate the PRS distribution, and we could then actually have a look to see whether our PRS was associated with those different cohorts and pull that information back. So, and this is work that is being done within my group by Anshika, and she's been pulling together the various stages. So this is kind of putting together what we've done, what we're doing so far, is that we have our PRS workflow down here, which I will cover in a bit, but we are then, so this is a slightly different view, that we query the beacon network to find data sets with our variants or variants of interest, and then we'd use the distributed authorization to get access to it, and if that was successful, then we can take the data to our staging area. And here we're using the EGA as our example, because this has been already set up for a lot of this infrastructure, and there we would be able to run our pipeline and extract the data. And the idea is that this is our kind of diagrammatic view of our workflow, that we're putting together lots of different modules that are all connected together. So this is using nextflow and Singularity really containers. We're building the different modules and linking them up and taking the data from the different targets, running, either going through association analysis or PRS, depending whether this is needed. We're going to add in extra things here for imputation if should that be required. And so it's going to be quite a modular system that will hopefully be expandable and easily extensible, but also potentially, actually, if we wanted to change the workflow entirely, we can just plug in new modules to make it work. And so at the moment I was saying, yes, it's modular. So these are kind of the standard QC steps that we put together in our workflow. And this is your standard Hardy-Weinberg, low genotyping rate, strands, things like that. And at the moment we've got a couple of bits on the PRS side of things as well. But this is still work in progress, and we will be adding more to this and trying to make it as generic as possible, because this is, as the whole point of CINECA is, this is a kind of a an instantiation of the theoretical things in the GA4GH. So one thing that has proved, I think, more interesting is obviously the whole kind of legal side of things, is that this is kind of across 3 different continents. There are different regulations, different national laws. In the EU, we've got GDPR, but GDPR is interpreted differently between the member states, but also different institutions as well have different views on this. And it's still quite a new area. So there's a lot of caution about how we can actually share data and how we can make data available and what we can do with this. And I think the key thing for us, and this was something that we really didn't have at the start of CINECA, is synthetic data and use of sort of synthetic cohorts. Because for testing federated systems, this means we're completely unencumbered by any pre-authorization. So when we're doing our distributed authorization, we can actually put in a fake loop that automatically grants access to anyone requesting that. So that allows us for fast testing. We can openly share it between all project participants, and should anyone take the data or there's any data breach, there is no risk of privacy being compromised. And we just need to make the data available by the same methodologies that we would be doing as if it was real data, and it should actually mimic very closely the real data. And there have been a number of cohorts that we developed for the different on different continents, in Canada and Africa and in Europe, and these are available and can be found linked on the CINECA web pages. So in Work Package 4, what we've really been using is sort of synthetic and sort of open access data as well. So have the Europe UK one, which is 1,000 genomes, plus, and we added phenotypes on this, and the Givardis RNA-seq as well. And then also the CEDAR data. And this has been used a lot for the EQTL workflow that is part of the Work Package 4, and is the one that we're kind of creating alongside the PRS. So this UK1 data set is based on 1,000 genomes, and this is something I actually worked on to develop for the Type 2 knowledge portal, to do testing across the different nodes to make sure there that our pipelines were consistent and we were getting the same results. And we needed a data set that could be openly and easily shared between all the participants, between UK and Europe, and this came up as the best option. And in CINECA, this has proved useful for our initial testing, but it's a bit small. It's 2,500 individuals for our PRS, and the whole point of CINECA is we want to, as we said in our mission statement, this is going to be sort of biobank population scale data. So we needed a bigger data set to actually test once we get up and everything working, to make sure that we can, our technologies will work at this scale. And we developed this new data set, this CIN500K, which is 500,000 samples and just under 27 million variants. We generated 10 synthetic phenotypes, which are continuous and binary phenotypes. And we're in the process of making this available via the EGA. So this is work that's been done by Andre in my team, and he's been developing this custom pipeline, and it's basically a number of custom strips and binary programs that he's all wrapped up into a singularity container. And we've basically been using a two-step methodology here. Is that initially we're kind of using coalescent methods to generate our initial data set. And these are generally, we can generate a completely anonymous data set. This is completely de novo, but it is slow in the development. And a lot of other methods have been using sort of resampling. So we are initially developing a 30,000 sample data set that we generate using this updated script, and it can take from 3 hours to 1.5 days per chromosome to generate. And in that, we were actually using recombination map to make sure the synthetic data structure had an LD structure that was more realistic and more like a real human genome. And then from that, we then actually take those and we resample that to then sample up to 500,000. And the resampling is much faster. And we're using a version of HapGen 2 that we've adapted to make sure that it's parallelizable. And this runtime then is 2 to 17 hours per chromosome. So that's a lot quicker to develop and generate these much larger data sets. So this then gives us a big biobank size data set that we can work with. At each step, we were looking for related individuals, which took about 5 hours, but actually we didn't actually use it because we didn't have any samples that we considered overly related between them. So, and then we generate the phenotypes using this phenotype simulator, which was adapted because otherwise it wouldn't cope due to the matrix size with the relatedness. So we've updated that, and that's a singularity container now. It can run through, generate data sets of whichever size with various phenotypes. And as I say, this one is now being uploaded to the EGA, and we're just going through the last bits of finalizing all the metadata, and hopefully it will be available very soon. So I think, what have we learned from doing this work, and what do we think the take-home message is? And I think that the one thing that really we never thought about was that synthetic data sets, we absolutely have to have them here for testing and the validation of the federated data systems. But we need realistic data sets of appropriate size. We've got to have phenotypes. We need the genotypes as well. And one of the key things that we found is that when we're generating these synthetic data sets, how do we say that the synthetic data set is any good? I mean, we can set up the phenotypes, we can run some associations, and we can do standard kind of QC metrics, but will it meet lots of other people's use? Do we have proper LD structure? Are there going to be other things that are an issue? And I mean, one of them is that, when we're doing an analysis, we don't tend to just analyze a single phenotype. If we're doing a type 2 diabetes analysis, we're adjusting for age and sex, and these need to be correlated. And if we're doing anything with BMI, then that needs to be correlated. And this suddenly increases the computational complexity of generating very large phenotypic data sets to go on top of these genotypes. So I think this is still something that really needs a lot of improvement. We're just starting to get some really good data sets of reasonable size, but I think there's scope here for a lot of work in really developing the data sets, developing the tools for the data sets, and also tools for validating the data sets. I know that I can pick up a synthetic data set from another thing. I can run my tools on it, test that I think it's going to be realistic enough for me to use, and then use it for whichever purpose I need. I think here, this is the other thing that is really important, and it's the data export from the trusted research environments. We need this. The European Health Data Space mentions this, and it's a question of how do you, if you're uploading your tools and you're running them, how do you then ensure that the data that the people are then downloading is fully anonymized? Now, you could either do it that the code is checked before upload, and I believe that's what Data Shield were doing, or you have to have someone checking every file that the users wish to download to make sure that that's the case. either way, that there's going to have to be some kind of validation of either the tools being used or the data sets being downloaded, and that's providing an overhead so that people who are providing the data and these environments are going to have people who are au fait enough with the data to be able to say whether this is anonymized and can be downloaded, or whether it still contains information. And I think because of this, these research environments are going to really need to be very flexible on how they work to make sure that they can accommodate uploading very, very different tools and potentially other data sets that people want to analyze alongside. And I think it's really going to be key in making these work is being really flexible in how you actually implement them. So one final thing here is, as part of this, and these were set up by Leslie Gass, who is a project manager on CINECA, we have a couple of working groups that we've set up as cross-project initiatives. One is the Federated Analysis Working Group, and again, we've had discussions and talks there from people at DataShield. But the idea is that everyone who's interested in this, it's an open discussion area, and we encourage people to come along and present. And I believe the next one is on the 17th of October. But these details are on the CINECA website, so if you do want to join and sign up, please do. And then we have another one on called the Joint Synthetic Dataset Initiative. And again, I think this is where we're trying to really get people together with similar needs in terms of requirement for synthetic data sets, and hopefully be able to generate and develop new synthetic data sets that are actually more realistic and just wider and broader in scope of the data that we can have. So I think I'm coming up on time. So at that point, I should acknowledge the funding here and just say thank you for listening, and I'm happy to take any questions you might have.